Good day everyone, this is Christine Oreneta, and for today, I will be discussing about enzymes. This chapter includes the following topics. First, classification and nomenclature of enzymes. Second, enzyme substrate binding, which includes the lock and key model, induce fit model. The third one is the Michaelis-Menten approach to enzyme kinetics. Next, the enzyme inhibition, coenzyme and apoproteins, and the last one is enzyme inhibition in the treatment of AIDS. Of all the functions of proteins, catalysis is probably the most important. In the absence of catalysis, most reactions in biological systems would take place far too slowly to provide products at an adequate pace for a metabolizing program. Enzymes, the catalysts that serve this function in organisms. Enzymes are biological catalysts that speed up the metabolic reactions that occur in the body. First, the classification and nomenclature of enzymes. For the nomenclature of enzymes, first, the trivial name. It gives no idea of source, function, or reaction catalyzed by the enzyme. For example, trypsin, thrombin, and pepsin. The second one is the systematic name. First part is the name of the substrate for enzyme, and the second part is the type of reaction catalyzed by the enzyme. This part ends with the suffix ase or ase. For example, lactate dehydrogenase. The third one is based on their EC number. Enzymes are classified into 6 different groups according to the reaction being catalyzed. The EC number is determined by the Enzyme Commission in 1961. So the EC numbers are 4 digits. For example, A, B, C, D, where A is the class, B is the subclass, C is the sub-subclass, and D is the sub-sub-subclass. The B and C digits describe the reaction, while the D digit is used to distinguish between different enzymes of the same function based on the actual substrate in the reaction. For example, for alcohol NAD+ oxidoreductase, EC number is 1111. Moving on, we have the 6 classes. First, oxidoreductases. It catalyzes the transfer of hydrogen atoms or electrons from one substrate to another. It is also called oxidases, dehydrogenases, or reductases. The second one is the transferases. It catalyzes group transfer reactions, excluding oxidoreductases. It is given in the general form A-X plus B, Bx plus A. The third one is the hydrolysis. It catalyzes hydrolytic reactions. It includes lipases, esterases, nitralysis, peptidases, or proteases. It is given in a general form A-X plus H2O yields X minus OH plus HE. The fourth one is the EC or the EC4 is the lysis. It catalyzes the non-hydrolytic removal of functional groups from substrates, often creating a double bond in the product or the reverse reaction. It includes the carboxylase and adolysase in the removal reaction and synthesase in the addition reaction. It is given in a general form A minus B yields A equals B plus X minus Y. The fifth one is the isomerases. It catalyzes isomerization reactions, including racemizations and cis-trans isomerizations. The last one is the ligases. It catalyzes the synthesis of various bonds coupled with the breakdown of energy-containing substrate. Moving on, we have the enzyme substrate binding. Before a reaction can be catalyzed, the enzyme and substrate must bind. The substrate binds to the enzyme in a special pocket called the active site. Binding to the active site is reversible and occurs through non-covalent interactions. So there are two models often used to describe the binding. First, the lock and key model. It assumes a high degree of similarity between the shape of the substrate and the geometry of the binding site on the enzyme. The substrate binds to a site whose shape complements its own, like a key in a lock or the correct piece in a three-dimensional jigsaw puzzle. So figure 1 shows the lock and key model. The second model is the induced fit model. The binding of the substrate induces a conformational change in the enzyme that results in a complementary fit after the substrate is bound. This model is a more accurate description of formation of the EX complex, as it explains how the binding of E plus S leads toward establishment of the transition state. Figure 2 shows the induced fit model. The third one is the Michaelis-Menten approach to enzyme kinetics. Michaelis and Menten developed a series of mathematical relationships to explain the behavior of many non-allosteric enzymes. The equation describes maximal velocity Vmax and the Michaelis constant, which is Km. Vmax describes the velocity of an enzyme-catalyzed reaction where there is a saturating level of substrate. The Michaelis constant Km is mathematically equal to the substrate concentration that generates half of Vmax. So a line-Weaver-Burk plot is a reciprocal plot of 1 over V versus 1 over S, and it can be used to determine Km and Vmax. The fourth one is the enzyme inhibition. Inhibitors are compounds that bind to enzymes and reduce the rate of catalysis. Inhibitors can affect an enzymatic reaction in two ways: the reversible inhibitor and the irreversible inhibitor. A reversible inhibitor can bind to the enzyme and subsequently be released, leaving the enzyme in its original condition. A reversible inhibitor reacts with the enzyme to produce a protein that is not enzymatically active and from which the original enzyme can be regenerated. So there are two principles of inhibitors. The competitive inhibitors bind to the active site of an enzyme and prevent the simultaneous binding of substrate. And the second principle is the non-competitive inhibitors bind to enzymes at a site other than the active site, but they alter the active site in such a way to reduce the catalytic efficiency of the enzyme. So the type of inhibition can be determined by using a Line-Weaver-Burk plot. So figure 3 shows the competitive and non-competitive inhibitor. Next is the coenzyme in apoproteins. Coenzyme is an organic non-protein compound that binds with an enzyme to catalyze a reaction. It cannot function alone but can be reused several times when paired with an enzyme. It also assists enzymes in turning substrates. So examples of coenzymes are nicotinamide adenine dinucleotide or NAD, the nicotinamide adenine dinucleotide phosphate or the NADP, the flavine adenine dinucleotide or FAD, Next one is the apoproteins are proteins which together with a prosthetic group forms a particular biochemical molecule such as hormone or enzyme. Example, polyprotein, which is a constituent apoprotein of a very low density lipoproteins. The next one is the enzyme inhibition in the treatment of AIDS. 3 key enzymes that are current targets of AIDS therapy are namely reverse transcriptase, integrase, and protease. First, enzyme protease catalyzes the processing of viral proteins in an infected cell. Integrase is needed for the virus to copy itself in the host cell. Without these proteins, viable virus particles cannot be released to cause further infection. Treatment of AIDS is most effective when a combination of drug therapies are used, and HIV protease, integrase, and reverse transcriptase inhibitors play an important role. The use of multiple inhibitors for the key viral enzymes allows levels of each to remain below toxic levels to the cell. That would be all, and I hope you learned something. Thank you.